Now we will discuss about the cell trafficking. It means what's going inside the cell, where from cell membrane to molecules transfer to endoplasmic reticulum and other sites within the cell. Golgi is very important part in cell trafficking. Golgi is distribution center for proteins and lipids from endoplasmic reticulum to vesicles and plasma membrane. Post-translational events in Golgi include modifying an oligosaccharides on asparagine, adding oligosaccharides on serine and threonine, and adding Mannose phosphate to proteins for targeting to lysosomes, usually for degradation. Golgi is basically the distribution center for proteins and lipids from endoplasmic reticulum and to vesicles and plasma membrane. And post-translation events in Golgi include modifying an oligosaccharides. It's on the asparagine, adding oligosaccharides on serine and threonine, and adding Mannose phosphate to proteins for targeting to lysosomes, usually for degradation. Endosomes are sorting centers. There are some sorting centers in the cell. These are endosomes, and they are sorting center for material from the outside the cell or also from the Golgi. They send it to lysosomes for destruction. These endosomes are sorting center for the material, like material come from outside the cell, or if some come from the Golgi, they send it to lysosome for degradation or back to membrane Golgi for further use. Eye cell disease or inclusion cell disease. Eye cell disease, it means eye is inclusion cell disease or mucolipidosis type 2. It is inherited lysosomal disorder that is autosomal recessive. Defect in acetyl 1-phosphotransferase. Failure of the Golgi to phosphorylate mannose residues, decreased Mannose phosphate on glycoproteins, leads to enzymes secreted extracellularly rather than delivered to lysosomes. Lysosomes deficient in digestive enzymes, and that leads to buildup of cellular debris in lysosomes. This cellular debris is also called as inclusion bodies. It results in inclusion cell disease. It results in coarse facial features, gingival hyperplasia, and corneal clouding, and restricted joint movements, and claw hand deformities, and kyphoscoliosis. Increased plasma level of symptoms similar to but more severe than Hurler syndrome, often fatal in childhood. Signal recognition particles or SRP. These are abundant cytosolic ribonucleoprotein that traffics polypeptide ribosome complex from cytosol to the rough endoplasmic reticulum. SRP is absent or dysfunctional. SRP leads to accumulation of protein in cytosol. When signal recognition particle or SRP is absent or dysfunctional, then proteins are accumulated in the cytosol. Vesicular trafficking proteins, COPI, COP2, and Clathrin. These three are vesicular trafficking proteins. COPI is Golgi and Golgi retrograde, cis Golgi, that leads to endoplasmic reticulum. COP2 leads from endoplasmic reticulum to cis Golgi anterograde. COP2 it steps forward anterograde. COPI step back retrograde. Clathrin is trans Golgi. Lysosomes, plasma membrane, endosomes, receptor mediated endocytosis. LDL receptor activity. For example, LDL is Clathrin, COPI, and COP2. So from plasma membrane there is vesicle, and there is coating on vesicle of Clathrin. Clathrin is coated on the vesicle, and they transfer the this this vesicle is transferred to the lysosome for further degradation. So this is Golgi apparatus. It has one is trans Golgi and other is cis Golgi. These are two sides of Golgi apparatus. So nuclear envelope and rough endoplasmic reticulum, these two are joined together, and there are some vesicles on the rough endoplasmic reticulum, and on these vesicles COPI is coated on these vesicles, and there are some other vesicles where COP2 is coated on these vesicles, and these vesicles are transferred to the Golgi apparatus.